Over the centuries, mankind has come into contact with many different afflictions caused by microorganisms, such as the bubonic plague, the Spanish flu, Ebola hemorrhagic fever, smallpox, and AIDS. However, we have yet to overcome the greatest pathogenic threat of the 21st century, and that is COVID-19. Starting as an epidemic in Wuhan, China in December 2019, the SARS-CoV-2 virus that causes COVID-19 has spread uncontrollably throughout the world, leading to a global pandemic by March of 2020. Over 2.5 million people have died, and over 108 million are infected worldwide. More than a year on, mankind has started to push back against the viral pandemic with a myriad of vaccines, drugs, and treatments. This is where my research in peptide-based vaccines comes into play. Even with effective drug therapies such as hydroxychloroquine, remdesivir, and anti-SARS-CoV-2 monoclonal antibodies, vaccination still plays an important role in the suppression of viral infection rates. Providing artificially induced immunity to a population at risk of the infection would drastically reduce the number of people who become infected, rendering the virus less likely to spread and thus saving lives in the process. Because of that, I am working on the development of a multi-epitope peptide vaccine that I hope would effectively immunize people against SARS-CoV-2. A peptide is a substructure that makes up a larger protein molecule. Some of these peptides have antigenic properties, meaning they can trigger the immune system to begin countermeasures against the viral infection. Different peptides can trigger different types of immune responses, such as antibodies that neutralize viral particles by B cells, and the secretion of chemical messengers by T cells that in turn alert other immune cells of the ongoing viral infection. The principle behind the multi-epitope peptide vaccine is using several peptides that each trigger different immune cell activities as a vaccine to prime the host's immune system against the potential SARS-CoV-2 infection. The work I do in identifying the SARS-CoV-2 epitopes is crucial to the foundation of this vaccine. The same can be said about verifying the immune response of each epitope so that the potential of each epitope can be maximized to the fullest extent in a final vaccine product. The damage wrought by COVID-19 has affected millions of people all over the world, and not just to their health. The quarantine measures taken to stem the number of infections has caused the closure of businesses and people becoming unable to make their livelihoods. This in turn has affected the global economy on a critical scale. With that being said, it is important for an effective and safe vaccine to be developed against COVID-19 and for people to become immunized against it. This is to ensure that a return to normalcy can be made as quickly as possible while minimizing the human cost.